Hi everyone, thanks for coming to my talk. I'm Yanting. Today I'm going to talk about exploring chemistry into heavy hydrocarbons, automatic fragment modeling. And this is the agenda for today's talk. I will start with the background introduction. So why are we interested in heavy hydrocarbons? One of the reasons is that it's the starting material for lighter hydrocarbons, and it will be useful for improving the current refining process or polymer recycling process. And instead of doing experiment directly, what's the advantage of making a kinetics model? So sometimes it might not be able to conduct experiments under certain conditions. And we can get a sense of the chemistry of a system of interest, which we do not have the data by hand. And what techniques can we use to build kinetic model? So there are main categories. The first one is the simplified one, called the lumped species. So we usually combine the similar species together into groups. And we usually lump based on what we can observe experimentally. For example, aliphatic species versus aromatic species. And the other one is the detailed one. So it instead track every molecules, including all the isomers. And here is the brief table trying to comparing both methods. So what's the reason to add all the extra complexity? The first reason is that it can save the effort to conduct experiment to get the fitted rate coefficient. And it can avoid the inaccurate extrapolations from the fitting conditions, as well as capturing the cross reactions with the rate estimated from first principles. So now I'm going to introduce one software which can help us to build kinetics model automatically. It's called Reaction Mechanism Generator, RMG. It's an open source predictive tool for reacting chemical systems, and it constructs comprehensive models consisting of elementary reactions. And it will estimate all the unknown kinetics and thermochemical data by either looking for the database from literature or quantum chemistry calculation, or perform the estimation by using the group additivity method. So here I'm going to show an example of ethanol oxidation. So the starting species will be put into core, and then they will react together to form some new species and new reactions. And all the new species will be placed in age. And the species in the core and age will be simulated at the desired conditions. So if some species exceed the characteristic flux, then they will be moved into core. So the whole process will repeat until it thinks all the important species is included in the system. So this framework is good for now, only for applying to the small molecules. As species gets larger, there will be combinatorial explosion in number of possible reactions and possible isomers. And currently, it will be still difficult to generate a fully detailed model for species larger than 20 carbons by RMG, even with memory saving techniques. But we propose a possible solution by maintaining species at certain size during modeling. Then it can effectively limit the possible number of species and reduce the memory requirements. So if we look at the right-hand side plot here, we can see as the carbon number in the feedstock increase, the total number of distinct molecules will increase exponentially. However, if we limit the species size with either 5 or 8 carbon numbers, then it can effectively limit the total possible distinct molecules. And then we can largely save the memory requirement and also increase the modeling speed. So the idea is that instead of tracking every species, we can instead keep track of molecular fragments that will behave similarly. So the idea of fragment is trying to limit the species size between the whole molecule and the tiny single functional group. And it will break down large molecules to reduce the computational cost, and then develop the reaction modeling between the fragments. So for example, if we start with this complicated molecule, then we will first determine the important functional group which we do not want to perform cut. And then between the groups, we can perform like the cutting position, and at every cutting position, we will attach the cutting label shown here. So this all species is called fragment, and the auto fragment modeling framework will basically try to generate kinetics model between fragments. This is the workflow of AFM. So it basically performs the same process as in RMG. It all starts with giving the input file with fragments specified, and then you can automatically generate the kinetics model between fragments. And after we having the fragment model, we can further like doing the post model generation process to either perform reattachment so that we can get the molecular information. For example, the molecular weight distribution at last. So how can we generate the fragments? Basically, it will use the pseudo atom, which is called cutting label, shown here, either R or L at each cutting position. And then we further define some specific pattern to avoid cutting at certain position. For example, we think aromatic CC bonds is really important, so that we do not want to basically perform cutting at the aromatic species, at the aromatic position. And we can also use the constraints to maintain the fragment size within user-specified range. So after defining the fragments input, then how can we do the fragment model generation? The first question we should answer is that how can we estimate the thermochemical properties of fragment? Since fragment itself is a pseudo component which has no clear definition of absolute values. However, we can instead estimate the change of properties before and after the reaction by creating representative molecule to obtain the fragment's thermal properties. So here is an example. For this fragment, we will generate a representative molecule and estimate the thermochemical properties of this species as the fragment's thermal properties. As for the rate constant, we basically try to follow the same procedure of RMG. So the fragment will match the reaction template and map to the corresponding node in hierarchical tree in RMG. And then it will estimate the kinetics parameters by using the experimental data or ab initio calculation data. So after finish the fragment model generation part, then we can further to try to answer that, what kind of information do we want from model? So we probably want to get to know either the reactant conversion or the carbon number distribution or the molecular weight distribution. But after reactor simulation, we only have a list of fragment concentration. Then how can we recover them back to the molecules? So now we have to do the final step, which is called the fragment recombination. And the idea is to categorize all the fragments into different classes. And basically, we will try to reattach every fragment with R with another fragment which has the cutting label L on the other side. So after reattach all the fragments in the system, we will have all the molecules back. So here I'm gonna to show some application examples. And first, we will try to use the simplest case, which is the long chain alkane. And here I use octane for example. So by comparing the fragments model with the detailed octane molecule model, we can see the reactant conversion is quite similar, and the carbon number distribution is also similar as well. But so it basically shows similar product distribution between the detailed model and the fragment model. However, there are still some difference in the molecular weight distribution. So we can know the cutting will certainly lose some connectivity information. And here I try to know like what's the difference between different initial cutting strategy. And here the system I use is try to use the phenyldodecan. So different color basically represent different initial cutting strategy. And here is the comparison. So we can see by comparing the reactant conversion, basically every model has a little difference, but the difference is within like only 1 order of magnitude. However, if we look at the toluene concentration, we can see the yellow one basically have a large offset. And in looking the molecular weight distribution, we can also see the yellow one shows a really different molecular weight distribution compared with all the other cutting strategies. So here we can see the cutting position matters, and it might miss some important pathway. And bad cut can lead to a really different model behavior. So at this point, the user should be very cautious about like what's the initial cutting strategy they try to use. And so what if we apply this to try to build a more complicated system model? So here I try to build a fragment model which contains 3 components showing here. So the blue lines are the fragments model and the green lines are the molecules model. We can see by comparing the conversion or the total radical concentration or the total functional groups concentration, we can see both models look similar. But if we compare the time spent totally, we can see there is a large difference. The fragment modeling largely reduced the time required to capture all the important chemistry for the systems. So, and what if we reattach fragments and compare them with the experimental results? So here, the red dots is the experimental result. And from here, we can see basically the fragments model can predict the important species in the system. And it shows quite similar distribution compared with the experimental results. So here's the conclusions. Detailed models can be generated a priori, and it's useful when collecting experimental data would be somehow slow or difficult. And auto-fragment modeling is now fully automatic, so the user just need to give the initial feedstock information and specify the temperature or pressure profile. And this fragment modeling framework shows potential to model many complex systems, either alkanes or alkyl aromatic species, or even multiple species systems, such as surrogate system. And this framework shows good model generation speed without sacrificing important chemistry. So for the acknowledgement, I want to thank my advisor, William Green, and my thesis committee, and all the other Green Group members. And thank you for listening. If you have any questions, please look at the link provided here or email me directly. Thank you.